Bismillahirrahmanirrahim. The muscular system. The muscular system is made up of over 600 muscles. The term muscle is derived from the Latin word musculus, which means little mouse. Muscle is a soft tissue containing protein filaments that slide past one another. Muscles have the ability to contract, producing movement or maintaining the position of parts of the body. There are 5 major properties of the muscular system. Muscles are excitable or irritable. It means they can receive stimulation and respond to stimulation from the nerves. Muscles are contractible. It means after receiving stimulation, they are capable of contracting or shortening. Muscles are extensible. It means muscles can be stretched without damage by the application of force. Muscles have elasticity. It means muscles can recoil or bounce back to its original resting shape and length after contraction. Muscles also have adaptability. It means muscles can be changed in response to how it is used. It will enlarge or undergo hypertrophy with increased work. On the other hand, it can go in atrophy if deprived of work. Types of muscle movements. There are 5 types of muscle movements. Straightening and extending of the joint to increase the angle between two bones or body parts is called extension. Bending of the joint to decrease the angle between two bones or body parts is called flexion. Moving of a body part around an axis is called rotation. For example, rotation of the head, neck, and lower limb. Moving of a body part away from the body is called abduction. Moving of a body part toward the midline of the body is called adduction. Types of muscles. There are 3 types of muscles. Let's discuss the cardiac muscle. Cardiac muscle is the striated involuntary muscle that forms the walls of the cardiac chambers, that is the myocardium. It contracts in response to signals from the cardiac conduction system to make the heartbeat. The visceral or smooth muscles. These are the non-striated muscles that lack a specific arrangement. These are the involuntary muscles which are triggered by impulses that travel through the autonomic nervous system. These muscles possess greater elasticity and is found in the walls of hollow organs or organ systems of the body. For example, the blood vessels, the respiratory system, and the digestive system, etc. Skeletal muscle. Skeletal muscles are attached to the bones. These muscles provide the skeleton with the ability to move by contracting and relaxing in response to voluntary messages from the nervous system. Let's discuss organization of skeletal muscle from gross to molecular level. A tough layer of connective tissue that surrounds the skeletal muscle is called epimysium. Within the epimysium are multiple bundles called fascicles, which are sheeted by a perimysium. Fascicle contain muscle fibers, which are encased with endomysium. As we discussed, muscle fiber is encased by endomysium, but it also is surrounded by its own sarcolemma, which is also known as the myolemma. Myo refers to muscle and lemma refers to membrane. Thus, myolemma is actually the cell membrane of a muscle fiber cell. Sarcolemma consists of cell membrane of a muscle fiber cell and an outer coat, which is made up of a thin layer of polysaccharide material that contains numerous thin collagen fibrils. Sarcoplasm is the intracellular fluid of a muscle fiber cell. It contains large quantities of potassium, magnesium, phosphate, and protein enzymes. Tremendous numbers of mitochondria lie parallel to the myofibrils. We will discuss myofibrils in a while. Sarcoplasm also contains glycosomes, which are granules of stored glycogen. The muscle fiber cell contains sarcoplasmic reticulum. The sarcoplasmic reticulum is a specialized type of smooth endoplasmic reticulum that regulates the calcium ion storage, release, and uptake. The rapidly contracting types of muscle fibers have specially extensive sarcoplasmic reticula. Each muscle fiber contains several hundred to several thousand myofibrils. The myofibrils have myofilaments. There are two types of myofilaments, that is the actin and myosin filaments. Myofilaments are large polymerized protein molecules and cause contraction. Let's discuss myofilaments in detail. Each myofibril contains two types of myofilaments. Each myofibril is composed of about 3000 actin filaments. Actin filaments are also called thin filaments. Each myofibril is composed of about 1500 myosin filaments. Myosin filaments are also called thick filaments. The side-by-side relationship between the myosin and actin filaments is maintained by a large number of filamentous molecules of a protein called titin. Titin is also known as connectin. One end of the titin molecule is attached to the Z disc, acting as a spring and changing length. The other part of the titin molecule tethers it to the myosin thick filament. Ends of actin filaments are attached to the Z disc. The portion that lies between two successive Z discs is called sarcomere. Due to the regular arrangement of myofilaments in myofibrils, the myofibril is composed of alternating bands, that is, the light bands and the dark bands. The light bands contain only actin filaments. These are called I bands because they are isotropic to polarized light. The dark bands contain myosin filaments as well as the ends of actin filaments where they overlap the myosin. Dark bands are called A bands because they are anisotropic to polarized light. The light bands contain only actin filaments while dark bands contain myosin as well as the ends of actin filaments. The alternating pattern of these light and dark bands result in the striated appearance of the skeletal muscle. The Z lines bisect the light band. A light band called H band sits within the dark band. H band is the zone of thick filaments that has no actin. Thus, Z line bisects the light band and the H band bisects the dark band. Within the H zone is a thin M line. It is formed of cross-connecting elements of the cytoskeleton. In the relaxed state, the ends of the actin filaments extending from the two successive Z discs barely overlap one another. In the contracted state, ends of the actin filaments overlap one another to their maximum extent. Also, the Z discs have been pulled by actin filaments up to the ends of the myosin filaments. Thus, muscle contraction occurs by a sliding filament mechanism. In order to understand how actin and myosin filaments interact to cause contraction, we need to study molecular characteristics of both actin as well as the myosin filaments. Let's discuss molecular characteristics of the myosin filaments. Myosin molecules form myosin filament. Myosin molecule, which forms the myosin filament, is composed of 6 polypeptide chains. 2 of them are the heavy chains and 4 are the light chains. 2 heavy chains wrap spirally around each other to form double helix called tail. One end of each chain is folded bilaterally called head. There are 2 free heads at one end of myosin molecule. 4 light chains are also part of the myosin head. 2 light chains are attached to each head. These light chains help control the function of the head during muscle contraction. The head has an actin binding site. It also has ATP binding site. Myosin head functions as an ATPase enzyme. This property allows the head to cleave ATP and to use the energy for the contraction process. 200 or more myosin molecules form the myosin filament. Tails of the myosin molecules bundle together to form the body, while the heads of the molecules hang outward to the sides of the body. Part of the body of myosin molecule hangs to the side along with head, providing an arm. The protruding arm and heads together are called cross bridges. Each cross bridge is flexible at 2 points called hinges. One where arm leaves the body and the other where the arm attaches to the head. The hinged arms allow the heads either to be extended far outward from the body of myosin filament or to be brought close to the body. There are no cross bridge heads in the center of the myosin filament for a distance of about 0.2 micrometer because the hinged arms extend away from the center. Let's study molecular characteristics of actin filaments. Actin filament is composed of three protein components: the F-actin protein molecule, tropomyosin, and troponin. F-actin protein molecule is the backbone of the actin filament. The two strands of the F-actin protein molecule are wound in double helix. Each strand of the helix is composed of polymerized G-actin molecules. Attached to each G-actin molecule is one molecule of ADP. It is believed that ADP molecules are the active sites on the actin filaments with which the cross bridges of the myosin filaments interact to cause muscle contraction. Tropomyosin. Tropomyosin is wrapped spirally around the sides of the F-actin helix. In the resting state, it lies on the top of the active sites so that attraction cannot occur between the actin and myosin filaments to cause contraction. Troponin. Troponin has three loosely bound protein subunits. Troponin I has affinity for actin. Troponin T has affinity for tropomyosin. And troponin C has affinity for calcium ions. Let's discuss transmission of impulses from the nerve endings to the skeletal muscles that ultimately lead to the interaction of actin and myosin filaments to cause contraction. Skeletal muscle fibers are innervated by large myelinated nerve fibers. Each nerve ending of the nerve fiber makes a junction with the muscle fiber near its midpoint called neuromuscular junction. The space between the nerve terminal and the fiber membrane is called the synaptic space or synaptic cleft. In the axon terminal are many mitochondria that supply ATP. Acetylcholine is synthesized in the cytoplasm of the terminal but is rapidly absorbed into many small synaptic vesicles. When an action potential arrives at the nerve terminal, it opens many voltage-gated calcium channels, leading to an influx of calcium ions into the terminal. In the presence of calcium ions, synaptic vesicles containing neurotransmitter acetylcholine fuses with the membrane of the terminal. The acetylcholine diffuses into the synaptic space and binds with a receptor on the muscle fiber membrane to open multiple acetylcholine-gated channels. The opening of acetylcholine-gated channels allow large quantities of sodium ions to diffuse into the interior of the muscle fiber membrane. The entry of sodium ions causes an action potential that travels along the muscle fiber membrane. The skeletal muscle fiber is so large that action potential spreading along its surface membrane cause almost no current flow deep within the fiber. Maximum muscle contraction, however, requires the current to penetrate deeply into the muscle fiber. This is achieved by transverse tubules. Transverse tubules are also known as t-tubules. They begin at the cell membrane and penetrates all the way from one side of the muscle fiber to the opposite side. T-tubules are actually internal extensions of the cell membrane. Therefore, when an action potential spreads over the muscle fiber membrane, a potential change also spreads along the T-tubule to the deep interior of the muscle fiber. The action potential causes the release of calcium ions from sarcoplasmic reticulum. This sarcoplasmic reticulum is composed of two major parts: large chambers called terminal cisternae and long longitudinal tubules that surround all surfaces of the contractile myofibrils. As the action potential reaches the T-tubule, the voltage change is sensed by dihydropyridine receptors. Dihydropyridine receptors are linked to calcium release channels in the adjacent sarcoplasmic reticular cisternae. Calcium release channels are also called ryanodine receptor channels. Activation of dihydropyridine receptors in T-tubules triggered the opening of calcium release channels in the cisternae as well as in their attached longitudinal tubules. These channels remain open for a few milliseconds, releasing calcium ions into the sarcoplasm surrounding the myofibrils. In the presence of calcium ions, the inhibitory effect of troponin-tropomyosin on the actin filaments is inhibited. The mechanism of this inhibition is not known, but one suggestion has been presented. Each molecule of troponin C can bind strongly with up to 4 calcium ions. The troponin complex then undergoes a conformational change and moves tropomyosin molecule deeper into the groove. This action uncovers the active sites of the actin. Before contraction begins, the heads of the cross bridges bind with ATP. The ATPase activity of the myosin head immediately cleaves ATP into ADP and inorganic phosphate. ADP and inorganic phosphate remain bound to the head. When active sites are uncovered, myosin heads bind with these sites of the actin filament. The bond between the head of the cross bridges and active sites of the actin filament cause a conformational change in the head. The conformational change prompts the head to tilt toward arm of the cross bridge. The tilt of the head is called power stroke. Once the head of the cross bridge tilts, the ADP and phosphate ions are released. At the site of release of ADP, a new molecule of ATP binds. This binding of new ATP causes detachment of the head. A new molecule of ATP is cleaved into ADP and inorganic phosphate to begin the next cycle, leading to a new power stroke. Thus, muscle contract without changing the length of the myofilaments. After a fraction of a second, the calcium ions are pumped back into the sarcoplasmic reticulum by calcium membrane pump. They remain stored in the reticulum until a new muscle action potential comes along. This removal of calcium ions from the myofibrils causes the muscle contraction to cease.